Hey, can you change course to 045? Thank you.
Looks like another sponge that fell over.
Right, I was trying to see if it was the same one, but I'm like, that's not the same rock.
Oh, small coral there.
There too.
Another one on top of the rock.
Yeah, interesting. Here now they're on the rocks instead of anchored in the sediment. But that one also looks dead, maybe?
Yeah, I mean, it's just leftovers.
Something over there. We'll be heading up slope shortly
Oh, and
too,
here's...
so. Yeah. Sponge graveyard.
I was just gonna say, wow.
Ooh, another little fish.
Okay, I have another question that just came in here, and so it's in, so do your instruments gather data related to dimensions, species, etc., or is it only visual data collections and your observations? So perhaps Josh or Kylie might be able to answer this question.
I could also take it too.
Yeah.
Oh,
Yeah, go for
okay,
it, Taylor.
Taylor, yeah, take it.
Yeah, so I'm the data logger, so I'm currently taking notes on everything that we're seeing. But not only are we taking observations and video footage, we will be, looking at eDNA, environmental DNA from the water samples that we take in, as well as doing some analyses on what these actual corals and things we are collecting are. Sometimes you can look at a coral and think you know what it is, but you have to genetically test it to see if it is a different species or not. So yeah, that is what we're doing out here. Characterizing this environment to see what species we're finding, if there are any new species, and in what densities and diversity we're seeing.
Thank you.
Yeah.
Awesome.
There's another coral down there.
It's a large dead
Big
sponge.
dead one, yeah.
On a knocked over planter.
No, I think that one's fine. I'm more worried about the Atalanta one. Yep, I think there's just that little, you're seeing the little tail that's left over on that second piece of tape. That one looks solid to me. That one does not look so solid to me. Yeah, I'm worried about that little
oh, no way.
extra piece of like, I'm not so worried that it's loose because it is tied off, but if that little sheet bend comes undone, that would pretty much spell the end of the dive. This one looks good to me though. Don't you think? You don't think so?
I'd prefer it to be in the tape.
Well, there's the last piece of tape and then there's a little piece of tail, right?
Okay.
Right? No?
Yes, I mean, I agree with you, but when you put the tape,
It's usually right at the end of it, so I didn't realize there was just a couple inches
Yeah,
left off.
yeah.
Remarkable how linear these ripples are.
Mm-hmm.
Continuous.
Yeah, I keep looking at them. So what about a flow, you were saying, Rob, would make these ripples?
Well, for these types of sediments, you usually need flow in excess of 15 centimeters per second.
Okay. And when you say flow, is that relating to the water?
That's the current.
The movement
yeah,
of the current? Okay. Cool.
But it doesn't have to be consistent. It can be, periodic or episodic to have these sorts of things.
Oh, okay. So they would remain there, say if it was a periodic situation and it just happened and then it stopped, they would stay there for a bit before they would ripple away, I guess.
I mean, we do see some, seasonal changes in the flow pattern in this region based on the models.
Interesting. I'm not...
I don't know what that is.
Okay, so I have another question that has just popped in for anybody who may know, have the answer is, have you seen any Xenophyophores yet?
Not on this trip, no.
Not
Not yet.
What are those?
What is a Xenophyophore?
It's a fish that looks like it's in a bad mood.
Oh, hey.
It's
Mr. Grumpy
red.
Pants.
And
It's a Mr. Grumpy Pants
short
fish.
body, big,
Oh, is it
big
frogfish?
face. Xenophyophores.
I'm going to look it up right now.
Yeah. Usually reddish.
Oh, okay.
Could be a type of angler.
Oh, yeah, I've seen these guys before. They have little beaties. They're like little nubbies.
Big mouth, bad mood looking fish.
Little Mr. Grumpy Pants. But he's so cute with his little mouth.
I know, I like them.
Aw.
Especially when you see a baby one.
Oh, like this one.
Looks like they can have some really bright red coloration, which is cool.
Yeah.
Hopefully we can see one of those.
Yeah. They are quite striking. Well, and the texture of their body, like their skin, and anyway, very cool. Good question. Thank you for asking. Oh, yes. Chana chops forever. Long live. Do any of the rovers have metal detectors?
You can put them on there, but we don't have any right now.
Oh, okay.
Magnetometers.
Magnetometers. Cool. Got that. Okay, got those answered.
We got another coral.
Oh, where?
Tipsy coral.
Center of the screen.
Oh, there we go.
Got a floaty.
Yeah.
Oh,
Another
it's a
shrimp.
Shrimp, yeah.
Yeah.
I'm not sure what that thing is.
I think it's another one, but maybe not.
another shrimp? Oh, yeah.
I mean, if you find a holothuria in here, you might be able to get sediment enough here.
Yeah. Although it still looks rocky. It's hard to tell how deep the sediment really goes, you know?
So Liz, now we're starting to see these patterns again. Are those suggestive of sea cucumbers? I was noticing that before, remember, they started to go away.
Yeah, potentially.
Now they're starting to come back.
Yeah, I think whether or not we're seeing them is probably more of a function of the current and how much the sediment gets turned over.
Okay.
Yeah, I guess it doesn't suggest to me that they're right in the vicinity right now, but yeah, it could be a lot of different things, but it's probably a cucumber that's making that. That would be my best guess, just based on the size and the fact that it's at the surface.
Right. Huh. Cool. Oh, no.
Missed it. Yeah, there's not much here.
No.
In this area.
Oh my goodness. That is a very cute crochet Shauna cup. Thank you for sharing. Those are very cute. Oh, look. Oh.
I'm going to need the link to that.
She just sent it to us.
That is so cute.
Yeah.
It's in the chat.
It's in the chat. There are very many things. Crocheting has come back. I used to crochet halter tops and stuff, and it was really fun therapy. And then now I'm finding that people crochet all kinds of things.
Yeah, bags, shirts. Yeah, it's amazing.
Shirts,
and now deep sea animals.
My one friend, she crochets little octopus, and they're really tiny and cute. So then you can just. She has like a whole reef system. They do coral reef. They make coral. Instagram. There's a whole crocheting community out there. Have you seen any trash this expedition? Taylor, I'll pass that to you because you were talking about debris.
Yeah. Was that yesterday now? That
I think it was, yeah.
Yeah, we saw this
right?
like spirally orange piece of material that definitely was not organic. Don't know what it was. But yeah, the 4 years that I've been coming out here, the 4 cruises I've been on, every single cruise we've seen a piece of trash at least on a dive.
Wow.
Maybe not every single dive, but every single cruise for sure.
Every single cruise. Interesting.
Yeah.
Good question. Thank you for asking. Oh, somebody said we also saw a flip-flop.
Yeah, I thought I saw that this morning when I was waking up, a blue one.
Oh, okay. Wow, interesting.
Oh, that's sad.
Thought it made it all the way down there. Good grief.
Yvonne, are you missing a flip-flop? Yeah, there's another one of those things.
Yeah.
Can you see what that is there, Josh? Do you see that?
That curly looking.
Looks like a little fan almost.
Yeah. I'm seeing a couple of those.
Oh, sorry, can you point out in the teletrator? Oh, there.
Weird. You guys have really good eyes.
Zoom in, please.
It's like a ping pong paddle.
It's a coral.
Whoa.
Is it? Is
I
it one
think
of
Nice
those
stalk, you guys.
calcified hydroids?
It looks like it. Let's see.
Did we want to collect those if we saw them? I think we did, right?
I think we have to see multiple though in
Yeah.
order to collect.
Yep.
Yeah. Huh.
Interesting.
Yeah.
That's very interesting.
Yeah, that's
Is
weird.
it just covered in sediment and we can't see like the webbing in between it, or is it just completely solid?
Yeah, I don't know. Maybe it could be dead, I guess.
Yeah.
Huh.
Oh, I've seen a couple of them now.
Yeah,
Go come wide, please.
I think this is the second one I've seen.
You can see the way it
Yeah, it
pushes
doesn't
the
look like
current
any of
around.
those.
Kind of takes
Yeah.
a little, excavates a little bit of sand out of there.
Surprised we're still pretty sedimented here as we're coming up this nose a little bit.
Hmm.
Somebody said it might be a xenophore, and we've seen a few.
Oh,
Yeah.
Xenophyophore?
Oh. Usually they're dark brown, no? Or are they? I guess they might come in different colors.
No, I think they can be light.
Oh. I haven't
Cool.
seen one of those. That's cool.
And the lady who or the person who crocheted the chana cups, she crochets all kind of deep sea critters because she has been inspired by watching our nautilus dives.
Oh, that's
Aw,
great.
That's
Thank
wonderful.
you.
That's awesome.
Can never spell Xenophyophore.
It is.
Start with an
right here,
X.
like,
Yep, I know that.
Xeno,
That's it.
Xeno,
Oh, there we go.
Yeah.
PHYO4.
the
Okay,
DVL
PHORE.
Oh, there is a
Sorry, what's
different
Can I
spelling.
just reset the DVL again?
Sure.
What is our depth now?
2330.
Oh, wow.
Yeah, 2340.
So we've come up a bit.
Yep. A little steeper slope now.
Okay. Okay, wait, does it look like we're on the side of it?
Yeah, we're
Or we
coming
just
up.
had
it-
it tilt.
It's a
Oh,
little
okay.
steeper.
I see. What is that? And another
Just a
sponge.
Big rock? Oh, I see.
It's a
Right
sponge
there
right
in the
there.
bottom
Yeah.
left corner. Oh, sorry. I don't have the little pen.
An anemone maybe?
Another sponge or something?
That's a, yeah, I think it's
Yeah, it
a
looks like a
sponge.
And then
sponge.
It's a
an
sponge
enemy
sponge.
down below.
Yeah.
Oh, and an anemone.
Can you zoom in, please?
Okay.
And a jelly?
Xenophore.
Oh, no, yeah, there's
A
the
little
jelly. You want to zoom on that jelly?
Oh.
Yeah, okay, we'll start with the jelly.
Party.
Sorry, too much to see. Oh, the jelly's gone.
Oh, dang it.
It's there, it's just gotta
There it
back
is.
up. There you go. Let me come up
Ben,
a little
is
bit.
that a bentacodon?
Okay, we'll zoom in.
The jelly?
Yeah. No.
I don't know.
Oh,
Uh-oh.
how cute.
Oh.
Oh.
Zooming around.
Put 'em-
Let's see.
all right.
It's really cute.
It is super cute.
Okay, we'll come wide, please.
Oh, there he is now.
Here we
No.
go,
Oh,
buddy.
come
Yeah.
back for another shot.
I think it
Little
could be a
cameo.
bentacodon,
All right.
but
Okay,
maybe
we'll
not.
come wide.
I'm
And
not sure what its living range is. I'm used to diving in different areas.
Mm. Yeah, I'm not good with jellies. But it could be.
Okay, we'll zoom in again.
Oh, with the anemone, a sponge and anemone.
Mm-hmm.
Okay, come away, please.
Oh, so we got a sea slug question. How many slugs? Oh, good grief. You read it, Rob.
I'm not going to read it.
How many slugs would a sea slug slug if a sea slug did slug slugs? I did it! Who did that?
4.
Did you break that?
Nope.
No. It's their website. Hmm? Oh, a pom-pom or a cereanthid anemone. Okay. Okay. So
Yeah, just a sedimented talus slope here.
Mm-hmm.
So now we're going back to, or would you call these larger nodules?
No, these are,
These would be pillows.
yeah, these are
Okay.
bits and pieces of pillows.
So in the chat box here, somebody commented that xenophores are usually sediment colored since they are kind of made up of sediment gooped together. And then another comment was we would expect to see life increase after the 2,000 meter mark.
Yes.
And then the anemone could either be a pom-pom or a cereanthid anemone. Those are some current comments in our chat box.
Sounds like a scientist.
Mm-hmm.
Not sure what it was, but I'm quite sure it wasn't a pom-pom.
No pom-pom.
Don't think so. They look like pom-poms.
They do. Now I want to see one. Oh, they do look like pom-poms.
Yep.
Cute. Are you seeing any good rocks here, Rob?
No, these are just manganese covered.
Oh,
chunks.
that was, yeah, so that was it. You said manganese covered, and you can tell that they were covered because it seeps into the little crevices of the rocks. Is that what
It kind of
you're
coats
saying?
the outside.
Coats the outside. Ouch.
bridge nav. You can extend the course a little further to 500 meters because we're going to continue on for a while. Thank you. I just didn't want you to have to do extra work.
I wish we did. So we have not seen a blue dragon sea slug yet, but that would be pretty cool if we did.
Can you zoom in, please, Dave?
That's the Portuguese where nudibranch I was talking about.
You see a star there?
Yeah.
And a coral.
Looks like Stitch.
Hey, Dave, can you zoom in, please?
This might be the first star we've seen just like on a rock.
Oh, wow.
But there's a coral next to it.
Making his way over. Start the climb.
Time
yeah,
to
it is.
predate. That's what they do.
It's a nice bamboo coral.
Yeah, come wide, please.
All right, Rob, we've got something here for you again. Do we know the mechanism for the iron magnesium crust deposits? Is it excess solute, biological, etc.?
Well, I think for the most part, people think it's, a chemical reaction, so maybe excess
Zoom in,
solute.
please.
But
Oh, okay.
they haven't been able to rule out any sort of, microbial interaction in some of these. They're still trying to see if there's some sort of possibility that microbes are involved in it.
Ah, interesting. And they're involved in, for creating the iron magnesium crust deposits?
Yeah, for
Wow. Interesting.
It'll zoom out.
So somebody did just ask if we have seen any chimaras. I'm not thinking that we have yet. The cartilaginous fish. Yeah, it doesn't look like we have yet.
I don't think you usually see them this deep.
Okay. Ghost sharks. The nodules are usually the size of texture we see in this one. Did you read? Nice. right, you guys, as we're traveling through this area, what kinds of things are you hoping to see as we get into the shallower depths of our cruise, of our dive?
Hopefully more corals and larger corals.
Yeah, huh?
Mm-hmm.
What types of corals do you think we're going to see?
Mm.
I know we've been talking a lot about bamboos, but there's some other ones that have been mentioned here in our chats. Let's go back to the science guys.
I know Steve is hoping to see some primnoids.
Primnoids.
Yeah.
Oh, here's another
Yeah.
fish.
Oh, hey,
Yeah.
a fish.
Oh,
Mhm.
Fly by.
So perhaps we'll start seeing some more fish too, huh?
Hopefully,
I'm
yeah.
hoping we
Moving
see some
along here.
Paragorgia. That would be, I like those ones.
Paragorgia?
Big,
Is that the
big
bubblegum
fans.
ones?
Yeah.
Yeah, bubblegum corals are very pretty.
Ooh, bubblegum coral, okay.
I like the yellow ones too.
Yeah.
Mm.
Wow, nice. I'd like to see some more fish. I'm curious to see what kind of fish are coming around.
Yeah, I want to see a Dumbo octopus.
Right?
Me too, me too.
They're a crowd pleaser.
Yep. Any octopus,
Oh,
really.
so chimeras can be at these deep and deeper for sure.
Oh.
Chimeras are often called abyssal ghost sharks, which is a misnomer because they don't live in the abyssal area, nor are they sharks. Ah, mahalo for that information.
Yeah, thank you.
Awesome. Oh, and then here we have how long is left on this dive. So we still have until about 9 o'clock tonight, which is a good about 8 hours, 8 to 9 hours, excuse me.
What,
Yay,
for
thanks
our
for-
watch?
No, on the dive
Oh, okay.
overall,
Yeah,
so.
I can't believe we're almost done with this watch.
I know,
It
we're
went
really
so
fast.
It's amazing.
Yeah, it really flew by.
yeah.
Mm-hmm.
Oh, somebody said that they love the iridogorgia coral, the firework coral, and I love the Poliopongon sponges. I want to see a Caulophacus sea spider.
Mm.
Ooh,
Those are
Oh
cool.
boy.
Scary.
not me.
Wasn't there a sea spider? I want to see one. Really?
Got another coral here.
Hey.
Yeah.
Oh, yeah, keep
Is that a
doing
spiral one again?
Go zoom in, please. No, I think it's, is it the spirally
Oh
one?
yeah.
Or is it
looks
the
like,
Chrysogorgia?
Oh, it
good point.
is.
Yeah, it's all bushy.
Bushy coral?
Yeah. It's
Yeah,
got its-
Chrysogorgia.
It's got its associate hanging out. This one's
Oh,
always
this
seemed to have
tight- this
one
keyboard is
little
so hard.
spot lobster in there. See him? Little claws. Okay, we'll come wide, please.
Do we want to turn the lasers back on just while we're exploring here? Thanks. Oh, there's a
Oh, here's another
anemone down
anemone.
there on the bottom left.
Oh, yeah.
You'll zoom in there too, please, Dave.
Having the telestrator is nice, but I almost kind of miss the where's Waldo aspect of things when someone says.
I really don't. Although we could go back to the stick.
Well, that's right.
Yeah.
You
We
need a
still
long
have-
one in this
We
van.
have it for posterity. It's in here. Okay. Go wide, please. That's right. I think it's behind you somewhere.
Somebody would like to see some siphonophores.
Siphonophores?
Thank you.
Yeah.
We saw some on the way
Siphonophores.
down.
Right? I think we were talking about that
Probably
last
see them
night,
on the way
yeah?
up again,
Yeah,
too.
stay tuned for the blue water.
Yep.
Oh, yeah, yeah, yeah. Too bad we can't see telestrator. Somebody's really pushing for that telestrator.
It's not that exciting
It's
for
not.
the.
No.
You're not missing much.
We end up looking right where we telestrated anyway. So.
Yeah.
We're just drawing up hockey plays for the Stanley Cup tonight.
Exactly.
Ooh.
Diagram the power play for us there.
Yeah. We sure what that is. Can we zoom in on that, Dave, please? What is
I
that?
didn't even see
Stalked
that.
coral something? Oh, is that a bunch of like zoanthids that have taken over?
Oh, wow.
Whatever that was?
Oh, yeah.
Maybe?
Yeah.
Yeah, come light, please.
Oh, okay.
So if we got a handover coming.
Okay.
Oh, we did.
Oh, someone would be
I don't
stoked
want
to
to cheer.
see the blue or bluish green
That
turquoise
would be inappropriate.
plexurids. Awesome.
Yeah, those are beautiful. I would love to see one of those too.
Really, yeah.
Another stalk.
Oh, here we go. Another spongy.
Hey, sunshine.
Oh. Caulophacus sponge. Thank you for sharing all of this information on our chat. This is awesome. It's really nice to see people engaging.
Thieves. Saboteurs.
Bye, you guys. Hey. All right. Thank you, everybody, for joining us on Nautilus. I'm going to transition out, and we hope you enjoy the rest of your day. Aloha, hui ho ma lama pono.
Hello, everyone. Nice to see everyone again on the 12 to 4.
Good morning, Michael.
Yeah, good morning. Good nooning.
Well, good news.
Good nooning.
We're the start of the morning and the start of the afternoon.
Yeah.
Someone has coffee today, hey?
Yeah, I just downed a coffee cook, so I'm like, hashtag not an ad. So I am primed and ready to go.
Yeah, I'm still learning how to make coffee in the machine downstairs, and I somehow made it very fragrant, yet not very tasty.
As a good Seattleite, I brought coffee with me onto the ship, but I have been indulging in, particularly for the midnight watch?
Wow.
Oh, there's some nice sponges. It's like one of the farrieds we've been seeing on and off, and a possible maybe bolosoma.
Those are bath scrubbing sponges, right? That's where I put my loofah.
The Farriidae is going to be, let me pull it up here, Farriidae. And then bolosoma is bolosoma.
We're moving right now, right?
Yeah.
How much do we have left? So, because I got almost a wrap on the 6/8.
Roger.
Just keep that in mind.
Yeah.
So how is everybody? Is everyone feeling a little bit more vivacious this noon?
Yeah, feeling much better than last night.
Oh, yes.
We're going to have some fun jokes come in. Did anyone bring any jokes like we were discussing?
Yeah, anyone do their homework?
Sorry.
I somehow forgot that on my way to bed at 4 a.m. Yeah.
That's always the excuse, isn't it?
So if you're just tuning in, we are now exploring a recently mapped unnamed seamount near Johnston Atoll in the Pacific Remote Islands Marine National Monument. So stay tuned as we kind of investigate the biological communities and volcanic formations found about here.
I do have a fish joke for us.
Oh.
It's even a tropical fish joke.
Oh my gosh, how relevant.
What advice did the father clownfish give to his son?
I don't know.
Stop clowning around.
Keep your friends close and your anemones closer.
Oh, I like it.
That's a topical tropical joke. Because they live in anemones.
What's that?
It's because they live in the anemones. That's a tier 2 clever joke.
Tier 2 clever.
Yeah.
Raj. I'm going to go ahead and push down in there a wee bit, please.
Zoom it in. About halfway.
Yep. It's a cool little hold fast that it has.
Got any more zoom there?
Definitely.
It's got freckles.
It does have freckles, that's for sure. Okay, full wide, please.
Full wide.
Hey Chris, you want to show us the dive path so far? So we're at waypoint 7.
Oh, I still forgot to bring my glasses.
Ooh, I'm going to guess 4 kilometers.
Ah,
Reg.
So about a vertical kilometer, right?
Maybe. I'm not sure. Sorry, Tim.
Oh, sorry. I didn't mean to talk over you there. We started at 3,200 and we're at 2,200 meters.
oh yeah.
Oh, I see what you're saying.
Yeah.
Yeah, that's correct. I'll hold off here for a minute. Ready, set, run.
Just before lunch, I was worried it was going to turn into a mud dive as they crossed that flat area, but we got right back into the rocks just in time for us to come on to watch.
I was very considerate of them.
Yeah, we got all the luck.
I was worried there might be biology.
Is there a way to confirm that the images I'm taking are like loading up correctly? Because I'm not seeing them in my little preview box like I normally do.
Do you mean for the sextant?
No, just the capture on Hercules' camera.
Oh, you mean the screen caps?
Yeah.
I forget where to check that.
I'm not sure. You could. Yeah, I'm not sure.
All right, I'll check it out. Thanks.
Yep.
No, yeah, you're good. So I have a question for the back row. I'm assuming that these things are the little branch-like objects that we're coming across are deceased corals. Are they now geology, or are they still biology?
Some of them actually may be deceased sponges as well. And with some of the corals, I would argue that they were still biology until they get completely covered in the manganese crust, because they're still going to support some of the like hydroids and other small benthos, even as just a skeleton. With the sponges, I would say geology, because particularly with the glass sponges, they have these silica-based skeletons that can become fused together, especially after they die, and they basically at that point become a rock.
Can we get a zoom on the sea pickle?
Zooming in.
So Liz was looking to sample one and then take a sediment core nearby, even if it's shallow, if that's possible. I don't know if we're in a position to do that.
Wide.
And I can't hear if you responded to me, sorry.
Oh, sorry, I was off SPL. We won't be able to get a core around here. This is way much too shallow to get a plug. But we can, we could scoop. Yeah.
I think they'll only want the sediment if it's possible to get a core, just to keep it isolated from everything else that's going to end up in a box.
Rog. Yeah, then I'd highly doubt we'll get a sediment core then.
Yeah.
Given the rockiness and the shallowness of this. We'll get some nice sextant cameras, or images of this guy. All right. Let's take one more picture of it. While we're getting everything set up, go ahead and push on in there again, please.
Zooming in.
I'll turn on the porch. Hopefully that gets you better lighting. Ooh. Gonna keep an eye on the sonar there.
Yeah.
Okay, full wide, please.
Full wide.
What did they collect in all these other ones? Cool.
Do you need bubble?
Yes, please. That'd be great.
If bubble wants to cooperate, there you go.
Off the fingers, that's not good. Well, let's see if it'll work for us. Gonna go ahead and fire up the suction there. And turn off the... number 6. Number 6.
There it is. Oh,
Then go to maybe like plus 70.
70?
Yeah. The big one.
Are we flushing right now?
Yeah, so we're currently flushing.
We good?
I'm not seeing any flow go through there. You want to go ahead and...
I'm seeing some, I think.
Yeah, go ahead and kick it up to 100%. I'm going to take a sample here of the sediment. See if we can get any flow. Yeah, I'm not seeing any flow go through there.
I think they're planning to slurp this one.
Yeah, no, we're just trying to see if we have flow going through our section. Go ahead and zero that guy out there.
Let me try T2.
Is either T6 or T4?
T4. Okay,
Right.
I'll try T4.
I'm pretty sure it's T6 though.
I think so too.
Cool. Back row, did they try sampling any rocks or anything that's big that they shouldn't have? Okay, zero that one out and tries T6 again.
Hey.
I'm seeing a note here that says one of the push cores was attempted, but it was not successful. And that's all the indication I'm seeing for a rock that could potentially be too big.
Roger. Yeah, come up a little bit there. Okay, well, interesting. This guy's actually kind of big anyway for the slurp. Maybe we can use whatever section we have to put him in the forward bin.
That was what Liz just chimed in from the lounge, and she was recommending that she thought this one would get stuck in the tube to just use the slurp to put him in a forward box.
I agree, yeah. Is there any space in the forward bins?
Yep, there's space in Lambda. Just wanted to note that one of the nodules from Omega did fall into that box yesterday.
Sure thing. Okay, you want to go to about 40% on the slurp there?
40%.
Thank you. All right. That kind of worked. Go ahead and open up that forward box.
Forward box.
Make sure I'm full rack back here. That looks great. We got a nice shot of the underside. Go ahead and open it up all the way. Thank you. All right. Go ahead and zero the section there.
Yep. Zeroed out.
Okay. Oh,
Oh
no.
no!
Roger, understood. Well, that's unfortunate. Go ahead and close up the bin. That was otherwise kind of nice. All right, we're going to get you another one, and we're going to not do that again.
That was unexpected.
Unexpected turn of events. I probably should have checked all my cameras a bit better. All right. Well, let's find us either that sea cucumber or another one.
So we have a question coming in about what samples are we taking? So maybe Kevin, you can talk about the geological samples we're taking, and Meredith, you can talk a little bit about the biological samples.
Yeah, that sounds good. Yeah, for the geology, we'll focus mostly on basaltic lava flows. And the main reason, that's what most of this is made up of. And then it's covered in manganese. Up, there he goes.
All right.
Free as a bird.
Open the box, open the box.
I know, right?
Look, there's his guts.
Actually, question. Yeah. Stand by.
Well, at this point, he already evacuated its guts.
So we don't want it?
Yeah. You can see him floating around.
Roger. Fine. Sorry about that, guys. We'll get another one.
As I was saying, for the Geology, we're mostly interested in lava flows, mainly because when they cool, they actually record, they start a radiometric dating clock, and so they record the age that they since erupted. The ferromanganese crusts that grow on top of it are also interesting for geology because they record long-term seawater conditions. They grow at a rate of something like 1 to 5 millimeters per million years. And so you can get in a 20 centimeter crust, 80 or so million years of ocean history that's kind of time averaged, which is very interesting as well.
So we're taking a few different types of biological samples. The first and kind of most prominent is going to be the physical specimens of corals, sponges, and sea cucumbers, like we just saw there. We were attempting to collect that sea cucumber. Corals and sponges are for taxonomy, to better understand what species are down here, and also to establish what we call a DNA voucher library, where we will be molecular barcoding all of the samples that are collected so that we'll have a DNA reference database to use for the other type of sample we're collecting, which is environmental DNA samples. So as we proceed through this dive, we'll be occasionally taking a water sample with the Niskin bottles for environmental DNA. And what that is we're able to filter the water and actually extract DNA directly from all the material we capture on that filter, and that gives us a snapshot of the whole biological community in the area. Now, the sea cucumbers we're taking samples of are for Liz, who's on board. Liz is going to be using them to do a gut microbiome study to better understand the food web in these deep locations.
Was that?
We're just following this ridge, yeah?
Yeah. Well,
Like, how high does it go? Or, yeah, how shallow does it become? Do we know?
Yeah, we're going to head to waypoint 7, then steep up the slope to 8, and we'll see if we make it to 12.
Well, we've got till sometime just before 9 tonight to make it to 12. I think we can do it.
Yeah, I agree. And it looks like we have room for maybe one more rock, so I was going to save it till we're close to the summit to get at least one summit rock.
Means we've got plenty of space for biology to go on top of all of those rocks.
Then more rocks on top of the bio.
Dot matrix.
I can still hear the sound in my head of a dot matrix printer. You know,
I hear it now.
Oh.
Oh my gosh. I almost feel like there'd be like weather report. Stop. Clear skies. Stop.
Single sideband.
Yeah. It's really funny how like advancements in communications, like a lot of it is just like staying within the narrow bands they're allowed to be in, because they don't want to step on the toes of any type of like navigational bands or anything like that.
Right.
So it's like they're like stuck between like 4 and like 6 gigahertz or something like that.
Mm-hmm.
I don't know. There you go.
Did we ever introduce ourselves as we started this watch? I don't think we did.
I don't think so.
Yeah, no, let's go ahead and go around and introduce ourselves. I'll definitely start. I am Onye, your Science Communication Fellow. Happy to be on board the Nautilus again for the second time. And I'll be tuning into your questions and kind of facilitating them out to the scientists and navigator and ROV pilots. So yeah, keep sending in your questions.
Hi, I'm Kevin Conrad. I'm a marine geologist in the University of Nevada, Las Vegas. Back here, looking at a ferromanganese-coated branch of some sort. But yeah, I'm happy to answer any questions on the geology and help with the science.
I'm Meredith Everett. I'm a biologist and geneticist with NOAA's Northwest Fisheries Science Center, where I also do a lot of work with NOAA's Deep Sea Coral Research and Technology Program. And I'm here to work with Steve on the deep sea corals and sponges and also process all those eDNA samples.
Pushing in.
Hey everyone, I'm Chanel Vidal. I'm currently a PhD student at Arizona State University studying microbiology of dry permafrost in Antarctica. And I'm out here doing an ocean science internship with the Nautilus, and I'm loving it. And if anyone has any questions about what it's like to get a degree in geology and then study microbiology in their PhD, happy to answer any questions.
Hey, I'm Chris Krasnowski. I'm here working as a navigator, and I'm also working on the high-resolution mapping system, which maybe we'll see here on the next dive or two. Other than that, I'm a PhD, or a postdoc student at University of Rhode Island.
You're not a student.
Well, postdoc, whatever. Whatever, I don't know, whatever I am.
Researcher.
Researcher at University of Rhode Island. And yeah, that's about it.
I'm sitting next to Chris. My name's Jess. I'm flying Herc, and yeah, from California originally. So really like the sunshine.
And then you moved to New York.
And
or
moved
Connecticut.
to Boston.
Oh no, that's right.
Not
Yeah.
Connecticut, yeah. This is all over the place right now.
Yeah.
But
There's significantly less sun there, I understand.
yeah, there's a wee bit less, yeah. At least in the winter.
Especially in the winter, yeah.
Yeah. All right, Michael.
Hello everyone, thanks for tuning in. I am flying Atalanta here right next to Herc, and I am from California. I'm a grad student at UCLA. And yeah, this is my fourth. This is a small number of times, like 3, I think, that I've been here. And I'll go until they kick me off.
Yeah. Are you a Bruin then at that point for UCLA? Is that the mascot? Or is it the bear?
I mean, it's a Bruin, which is a rare.
Roger.
Yeah.
Berkeley's the Bears, right?
Berkeley's the Bears, that's right.
Yeah, the bear, yeah. And then you have UCR, which is a bear, Riverside, UC Riverside, which is also a bear, but it's set up as a Highlander, so so it's a bear in a kilt playing some bagpipes. So
really?
Yeah.
Solid.
That's what the student body chose.
How about you there, Tim?
All right. My name's Tim Dwyer. I'm sitting in the video engineering seat, where I manage the video feeds, try to keep the camera in focus, and also make sure that we're recording all of these awesome images that we can bring it back from the seafloor and study it later. And I am from Friday Harbor, Washington, where my day job is as a high school science teacher.
I'm doing a DVL reset when you get a chance there.
Tim, what subject are you teaching? Science subject.
I work at a small high school, so I teach lots of the science subjects. So biology, chemistry, physics, oceanography, marine biology.
What's your favorite subject to teach?
Believe it or not, and I say this without any irony, I love teaching Algebra 1.
That's the gateway.
Exactly. You can just make students' lives, like, watch their faces light up as they get the math.
As they get it,
And that you're
they
right.
come online.
They're like, I made a line with math. Like, yes, you did. And that, you're right, that's the gateway.
Got some really nice primnoid corals coming up straight ahead.
Oh, I'm at the end of my leash pretty much over here. Gonna give me a little bit of rope there.
Yeah.
We'll see him from afar.
I can get you. I can get a little bit. I got nothing in my way.
Reg.
Not gonna keep me down.
Go ahead and push on in there a bit, please. It'll be a little bit bouncy.
You are out of my scope, though.
Pull wide, please.
Pull wide.
I'll come back over to you now.
Yeah, you just need a lateral. Oh, I should probably do gauges. I'm gonna still bubble for a second, okay?
Roger that. Okay, there has to be a sea cucumber around here somewhere. I think.
Those ripple patterns are absolutely gorgeous. How did this form at the bottom of the seafloor?
From deep sea currents. So the currents can move pretty strong enough to reshape the fine grain sediments in the area.
Is there a way to look at ripple patterns in millions of years old rocks and see where the flow direction was going?
Yes. Yeah, in well-preserved rocks, yeah, you can find what's called cross-bedding, and it's essentially bedding that's derived from ripple marks, and you can reconstruct paleo flow directions from it. These sediments are mostly carbonate, and carbonate's actually not stable at this kind of depth and oceanic conditions. That's kind of why you don't see coral fossils here, because they'll dissolve. And a lot of this sediment is likely dissolving as well as it's being deposited, so you tend not to get a big, thick layer of it. And then, since it's deep in the ocean, it's hard to get it then onto land, which for these kind of seamounts, it just gets subducted.
Will all of the seafloor get subducted at some point? What is the oldest age a seafloor could be?
Currently, the oldest age that's in the oceans is 180 million years old, and that's in the west of us, kind of in the towards the western Pacific, not at the far end, but towards it. And that's about as old as you can get typically. Mostly by that time you're subducted. You can have what's called ophelites, which is essentially where sometimes the seafloor doesn't successfully subduct and instead gets pushed up onto land. And so you can have seafloor basically abducted onto land. That can be quite old. Most of like the California coast and Oregon coast are seafloor that was pushed up onto land and didn't successfully subduct, for example.
Seemingly the way biology always works. As soon as we get out into the beautiful sediment where you would expect to be able to find a sea cucumber and maybe take a push for, they utterly vanish. Oh, I did the classic thing and left my microphone up. Now that we've gotten out into the flatter area with more sediment where you would expect to find a sea cucumber, they have utterly vanished. And it's Murphy's Law guarantees that as soon as we're back into the rocks where they'll be harder to sample, they'll appear again.
We'll take them anywhere. I have a vendetta now.
There's still a decent amount of corals and sponges just on these loose singular rocks, kind of isolated.
Yeah.
Looks like we're coming up on something up on the left.
Bamboo coral on the left?
Bamboo, yeah.
Okay, thank you.
Start coming up a bit there again. Do you think he's correcting to 045? Yeah.
Why do we see iron and manganese on the basaltic rocks and none of the carbonates?
Well, depends on the rock type. So the carbonate rocks you can find, like when we get to a geo and it has a carbonate platform.
Nice.
Oh, it's okay. They can be covered in ferromanganese, and actually it's really hard to tell sometimes if it's basalt or if it's carbonate. But for a lot of things like corals in this area, the deposition rate of ferromanganese is on the order of a millimeter per million years, and the dissolution rate is, I don't know, but faster than that. So they'll probably dissolve before being coated.
There's sea cucumber there.
Got some sponges on the right.
Did anyone from the previous watches collect any of the Ferrea sponge?
I just looked on the sample sheet and they only indicated sponge, so I'm not sure what kind it was.
Ugh.
Oh, wait, actually, stand by.
Reg.
I wonder what this would sound like.
What was that?
Got another nice coral coming up.
Point. Branch bamboo coral.
Got a few things in here. There's a couple little chrysogorgids.
Good. Do a partial
Those are
zoom,
these,
please.
the puffballs.
Partial zoom.
Let's get there.
And there's a couple bamboos.
And bamboo.
And I think there's a little further forward of us, I think there's a primnoid as well. So nice little cluster of multiple species.
Do a little wandering around. Some partial wide there.
Partial wide.
Where'd the primnoid go?
It's up to the right.
Upper right. Oh, I see. Down below.
I've got the still camera on, so I'm getting a slightly more straightforward view on one monitor back here.
Roger. Go ahead and push on in there again, please.
Zooming in.
Full wide, please.
Full wide.
There's a larger one there. Is it dead Ferrea?
just as an update on the corals previously collected, I have here that they collected Chrysogorgia colonies, some bamboo corals in a Niskin bottle. There's some fragments of dead sponge found in a grab. Let's see, 2 to 3 visible sponges in a scoop, and there was a white sponge collected. The genus of that one is Semperella, and there was another Niskin bottle with, I think, 5 corals, one of them being Chrysogorgia, another one being a bamboo coral, and then another name I can't quite read.
Great, thanks for the overview.
There's a good sponge stalk laying there on the bottom.
Yeah.
So Kevin, you said you wanted to take another rock sample at the summit of the seamount. Is there a particular reason why you wanted to wait for that that specific area?
Yeah, just to get a better idea, some nice sponges, of how long these volcanoes take to make. So typically they take around 5 million years to grow. So I usually like having stuff from the base because I'm interested when they first start growing. But in some cases in this region, we find 10 million year differences.
Could do partial zoom, please, Tim?
From some lava flows taken from different parts of the seamount. So some of these might be really long-lasting volcanic structures.
Full wide, please.
Two different glass sponge species there.
Are they called glass sponges because of the silica gelatin you were talking about earlier?
Yep, that's the reason.
Deduced.
Their skeleton is made of literal glass, and actually, when you collect them, they're one of the things you have to really make sure you're properly wearing your PPE in the lab, wearing your gloves, because if you handle them without gloves, it can be like handling fiberglass with bare hands.
Oh, that's the worst.
Go ahead and do a quick zoom, please.
Quick zoom.
These ones are really cool.
Whoa.
Full wide, please.
Full wide.
Steve's given us an ID on this sponge is Caulophacus.
So back row, just would you mind giving us a lowdown of what are on the to grab list? So we want a sea cucumber. Do we still want that sponge sample? Sorry, that was not very specific. I think it was Riftia, or not Riftia, the one we were supposed to collect last night.
That farried?
Farried, yeah.
I think we could probably get one if we see another of that type. We might have come out of its depth zone, however, because the ones we're seeing up here are a slightly different type.
Roger. Anything else besides one rock on the collection list?
We're looking kind of for any unusual primnoid corals or plexaurid corals, which we're just starting to see some of the primnoids, so we'll keep an eye out.
Okay. Come down a bit on the delta, please.
So now starting to kind of crest a knoll here, so it's going to kind of flatten out. We should get some more sediment. Then we're going to head eastward for a little bit and start going back up a larger cone.
It'll be interesting to see how the biota changes as we go up this other cone, because we're going to change the aspect of the slope that we're seeing, and the current should be moving over it slightly differently, so it's an opportunity to maybe see a little different set of biology.
Oh, look at the trails here.
It's neat that you have sponges basically in soft substrate here.
If I remember correctly, that's for the anchoring basilia, is that correct? We can take a peek. They have like little hooks that they anchor themselves in the sediment with. Gonna go ahead and push on in there a bit, please.
Zooming in.
We'll go straight for the base.
That is wildly cool.
Yeah, look at that.
If you look at those under SEM, they actually look like little anchors, the ends of each of these little fibers.
Like almost like basically a root system.
Yeah, kind of. We'll do one peek up. If you want to push on in there a bit more, please. Cool texture.
Yeah, that lattice is
That
amazing.
lace lattice that you can see over those oscula is often one of the defining characteristics to distinguish taxonomy in sponges like this. And also one of the things that gives the glass sponges, the hexactinellids, their name, they're like these little
Partial wide,
six-spike
please.
umbrellas when
wide.
you look at them under SEM.
Very cool. All right, full wide.
Full wide.
We have some questions coming in about the sponges. Is there a certain method to kind of date how old these sponges are?
That's kind of a tricky question. I think you can do some aging on some sponges using radioisotopes. Otherwise, they're not that easy to date because unlike some of the corals, they don't form like any kind of annual growth rings in general. So you'd have to do isotope-based dating for most of them.
Cool. And how about like the size of the sponge? Is there any way to estimate like the pace of the sponge growing?
So in some areas, we've been able to estimate kind of the growth rate and size of sponges when we have done ROV dives on areas that either were previously affected by trawling and have since regrown, or sponges that are growing on shipwrecks where you have a known date where the ship sank. And so sponges can only be as old as that date. And so from that, we've actually seen some glass sponges can be fairly quickly growing. But to get into really massive sizes, they do take quite a long time. And in some parts of the world, like up in British Columbia, you can actually have true reefs that are formed as thousands and thousands of years of sponges growing over the top of glass sponges in particular, growing over the top of one another. Nice little fish there.
You can see all these tracks
Yeah, huh.
on the bubble cam.
So cool.
It's interesting here, you'll notice that there's no ferromanganese nodules in this sediment pile. It seems like they only form in the small pockets on the ridges and not on the big expanses over here.
That's a good point.
Kevin, do you have any hypotheses as to why?
No. I guess it has something to do with the fluid flow being too restricted or not restricted, but there are people at URI working on that now, some grad students, and it's up for them to figure it out. Smarter people than I. You can occasionally see sea cucumber tracks in the sand over here, little white tracks, but I'm not seeing any sea cucumbers.
I think the friend that we tried to collect sent out the word to the rest of them. They're all taking
Must
a
have been
hiding.
it.
I don't know what you're talking about.
That was your last. That was your one shot, Jess.
And the whole ocean, there are no more.
Got a really nice coral that's really hard to see, but just in the bottom left corner.
Vertigorgia?
Yes.
Go ahead and push on in there a bit, please.
Zoom it in. About halfway.
Can I turn on the porch light there a bit?
Porch. Nah, was oh, helps a little.
Go ahead and push in a bit more on the polyps there.
That's beautiful.
full wide, please.
Full wide.
This is Iridogorgia. That's IRI. Yeah. And another primnoid straight ahead.
Bridge nav, 50 meters, 050. That's 50 meters, 50. We're gonna start poking around the corner here.
Yeah, sounds good. Hey, back row, when we're going across that flat top, that's going to be what, like a 500 meters or so? Do you guys want us to increase the speed? Or would you rather
I think
keep
we
at this
might
pace?
look for a sea cucumber first, since I'm expecting it to be sedimented over here, so we might be able to do both the core and the sample.
Okay.
But as soon as we get one, yeah, we can speed it up.
Sure thing. Oh, there's one there. Is there a certain type?
I think any that are collectible and of a good size.
Okay, so do we want to stop here?
Yeah, let's do it.
Okay. Bridge nav, hold position.
Yeah, right before the swing kicks in.
Yeah.
Bridge nav, 30 meters, 240.
Go ahead and push on in there, please.
Zooming in.
Does this one have enough stuff in its guts? Right, they're doing gut content analysis?
Yeah, I think so. It looks like on the bottom. This is a full gut right there.
Oh, you're soft. I don't know if you're on.
So it's a go? Correct. All right, full wide, please.
Full wide.
I'll throw out the bucket cam there too. This one might be small enough to fit into the slurp.
Want me to flush?
Yeah, go ahead and start the flush. Back row, do you guys want us to put this in the slurp if we can? We'll do a measurement once we're out there, but trim off the hose clamp, but.
Yep, that works.
All right, go ahead and zero out the flush. Go ahead and push on in there a bit, please, video. A little more in.
It's like perpetually and like out of focus.
Thank you. All right. I thought it was just me. But yeah, it's kind of a blur on the ocean floor.
All right, go ahead and line up. What jars do we have open right now?
jars 7, 6, and 5.
Roger. Go ahead and line up on jar 7. I think this will fit all right into the hose here.
Oh.
6 works. 7 works. All right, go ahead and do 70% suction.
70%. There it goes.
There she goes. Now try for 100% full wide, please.
Full wide.
Oh, okay. Go ahead and 0 it now. In jar 7. All right. Thank you, Chris. Go ahead and line up on the flush and jostle it around in there so it settles nicely.
What sample number is that?
Sample 016.
Roger.
All right. It's such a satisfying squeeze when it goes into a tube.
Yeah.
All right.
I'm just having fun looking at the vortex on the report camp.
Making our own little mini hurricanes underwater. I'm ready for you, Chris.
All right. So do we want to continue to waypoint 7, which we're basically already at, or do we just want to go to the next, start heading to the next?
Yeah, I think we can speed along to the waypoint 8, or at least until the slope picks up.
Okay. Heading to waypoint 8.
Did you still want to do the sediment?
080.
080.
Do we want to do the sediment?
Yeah, so there won't, this isn't coreable, this is quite shallow, the sediment layer, so we won't get a plug on it, so I would highly doubt that we would get any sample for a sediment.
Yeah, I don't think it's worth trying.
Yeah, if there was like a nice valley where there is a bunch of sediment, then go ahead, Chris. That'd be useful, but I don't think we're gonna get a good plug right here.
Bridge nav, 750 meters, 080.
Which is kind of funny because it's a bit deceptive because you see sediment here on top, but it's actually just a very thin layer.
Yeah, and taking the coil, you're going to lose the top 4 inches or so just from, gravity as you try to take it out. Mm-hmm. So if you only have a couple inches, it just doesn't work.
Yeah.
We are going 808.
Mm-hmm.
No, 080.
I'm like...
Yeah, I was like, he's just having fun with us.
I'll start reading bearings out in radians later.
Oh, so helpful.
Yeah, I did the calculations in my head, thank you.
We'll end the cruise in quaternions.
Oh. Oh dear. So anyone else want to be nav? The only prerequisite is that you read in degrees.
We can go in parsecs.
Points? There's all kinds of good units.
There are.
Furlongs.
My favorite scientific unit is the spharedrup.
What unit is that for?
It's oceanographic, and it's, I think, cubic meters per minute cubic meters per minute. It's a volume over distance measure.
It's a flow?
Yes.
Interesting. Named after the oceanographic professor, yeah?
Yes.
Wow, to have a unit named after yourself, that's pretty cool. I went.
We're starting to get. Wait, no, that's the. Never mind. I mean, we still have a wrap, but it's not as bad as I thought it was.
Yeah, it's on a 6-8. As long as they don't, as long as they're like not in the same direction, it should be all right. We'll take it out when we go the opposite way. There is a bridge by MIT that has smoots. So it's the bridge's length is counted in the body lengths of one of these students from, I don't know, 1960 or something like this. So the bridge is measured in smoots. And I think it became a standard unit of measure, which is, I don't know, pretty funny if you're just doing this as a college gag, and then now it actually becomes a useful unit.
So I actually bring this up in my physics class when we're going unit conversions, and Oliver Smoot later on in life became the head of, I think what was the Professional Association of Standard Setters? I forget the name of the group that sets all the standards and measurements, but he was the head honcho of that later on, sort of predestined, I would say.
Oh.
Oh, the Von
Foreshadowing.
Yeah. 5 feet 7 inches plus 1 ear.
Plus 1 ear, yeah.
Just double checked the sphere drip. It's 1 million cubic meters per second.
Huh.
1 million cubic meters?
Ocean scale.
Per second?
The ocean's big.
No, the per second part is confusing me. I thought it was over.
That's still like a lot, though.
Would that measure something like the Gulf Stream flow, or like a major
Yeah,
current?
it's usually like large currents.
The Amazon River is about 0.2 sphere drops to give us size.
Oh, dang.
I cannot believe it.
That is a big wave.
Yeah.
Where's the largest current?
I think some of the largest currents, well, you have to look at it whether you're talking speed or volume. But some of them occur in places like some of the fjords are off like British Columbia in terms of speed. And then in terms of volume, you're probably talking about some of the big internal waves in the Pacific.
What's an internal wave?
So an internal wave is, they occur at the boundaries between two density layers in the ocean, and their movement between those layers, usually tidally forced. And that's a special thanks to Emil, who answered that question for us late last night on our last shift.
Glad I could forget so we could all hear it again.
So if you're just tuning in, we are currently exploring a recently mapped unnamed seamount near Johnston Atoll in the Pacific Remote Islands Marine National Monument. So go ahead, send us your questions.